Well, hey AP, hope you're doing well when you watch this video. So this is me going through chapter 4, the first part of chapter 4. I have already posted organelles on topic 4, a video I made a year or two ago, as well as some resources there. So this is just me going through the first part of it. Okay, so let's go ahead and get started. We've covered some of this in class already, but I just wanted to go through it with you again. As I told you, we've got to catch up a little bit. So we're probably going to do a little reverse classroom possibly at some points, and also me doing these videos so you can do notes at home. So compartmentalization, yes, it is a thing. Of course, just a biblical worldview is the gospel is not a little compartment of our lives. The gospel invades all of our life. Following Jesus demands everything, and surrendering to him in all aspects. And it's the best life. It's the best life possible. So here's some of the objectives that we have been talking about. And so cells are definitely the smallest unit of life. Detailed study began in the 1800s. 10,000 roughly different molecules. Crazy thing about here's some, the cells here, white blood cells from the looks of it. And so the cell theory, I would definitely know the cell theory. Knowing that cells are fundamental units of life. Cell biology is common in college if you so desire to go into the science route. All things are composed of cells, living things, and all cells come from pre-existing cells. That's the one that makes it a theory, because where did the first cell come from? Something science cannot answer. Cell biology is life, and life is continuous. So this is a huge learning objective when it comes to this unit. Cells want to be really, really small. Why? Because they want a ton of surface area to volume. They want a lot of space for materials to enter and leave, but not much volume for that. So they want a high surface area to volume ratio. And we'll practice some math with that here in a second. Well, not in this video, but in class. And so volume goes up a lot when a cell gets larger, okay? And so surface area determines the amount of substances that can enter and leave. So heat goes up as the cell gets larger because metabolic reactions increase. And so with that being said, that's perfect timing when it comes to a cell phone call. I am not stopping the video. I'll just pause it. And so you can see here that it's definitely something that we have liked it. But cells are small because they want a lot of surface area to get material in and waste out and not much volume. Okay, because as cells grow, volume increases way faster than surface area, and so that leads to problems not only from a heat standpoint, but from a material exchange standpoint. You will receive all these formulas on the test, any test I give you, and also any test College Board gives you. We'll practice this math in class as far as surface area to volume ratios. There is a lab you can do with this. We might be able to get to it, but we definitely will be getting to diffusion lab and cellular. Got to do an enzyme lab too next week. And so, cell membrane. So we're going to start talking about the cell. Cell membrane. All cells have a cell membrane. Without a cell membrane, a cell is not a cell. And so the evolution of a cell membrane is crazy important when it comes to the evolution of cells. Of course, it's very hard to explain how a cell membrane correctly when it surrounded all those living components necessary for a cell to exist. So selective permeable membrane, communication, and binding and adhering. Those are the cell membrane functions. Prokaryotes, no membrane-bound organelles. Eukaryotes have membrane-bound organelles. It's important that you know membrane. Don't just say organelles, because prokaryotes have ribosomes. They make proteins. Different microscopes, light microscopes and electron microscopes, transmission ones, 2D images. They have to kind of cut it in half. The specimen, here's a mitochondria. Scanning ones kind of show 3D images, which is pretty cool to see. And so here's some more objectives. Prokaryotes versus eukaryotes. And membrane-bound organelles evolved from previously free-living ones, which we're going to talk about that in a second. And so in the origin of the cell theory, why do biologists focus on the origin of the cell membrane, and what advantage does eukaryotes have over prokaryotes? We went over these answers in class, but if you don't remember them, I would pause the video, go through these questions. Okay, back from the pause. And so without a cell membrane, you don't have a cell. So the origin of the cell membrane is crucially important. Not only did it have to evolve and stick together in a really chaotic, toxic environment, but it also had to surround components necessary that could divide. What advantage does eukaryotes have over prokaryotes? That's all about compartmentalization and specialization. Eukaryotes have specialized compartments where a lot of different reactions can take place, okay? And so prokaryotes thrive almost everywhere. I mean, places that are too acidic, too salty, too cold, too hot. they're there. They're there. And so prokaryotes really have mastered the ability to live everywhere. There's more in a handful of fertile soil than the number of people who have ever lived. Of course, prokaryotes, here's a general outline of a prokaryote. Plasma membrane, so all cells have a plasma membrane. They do have a cell wall. It differs than in plants. And then a capsule is like an advantage, an evolutionary adaptation to allow survival to some species. You have ribosomes, cytoplasm, flagella, and some species is pretty cool. So some are disease-causing, like tetanus, tuberculosis, anthrax, gonorrhea. Some decompose, very important to the environment because they return nutrients back to it. Biotechnology, hey, diabetes used to be a death sentence hundreds of years ago. They're not now. Why? Because we make insulin. How do we make it? We use bacteria to make it, or yeast. I think it's bacteria, actually. Yeast would be something else. But we put the gene inside of a prokaryote and let it grow it, the protein, and then we purify it and put it into medicine. Lack of membrane-bound nucleus, structurally smaller and simpler, yes, still crazy complex, but we don't have membrane-bound organelles. Prokaryotes are placed into bacteria and archaea. Archaea means literally the ancient ones, so they're extremophiles, which means they live in extreme places. Domains are similar, but they are biochemically different. Prokaryotes have DNA, but it's not inside of a nucleus, it's inside of a nucleoid region. And then the rest of the cytoplasm consists of cytosol, which is like dissolved material, and ribosomes. So that is an organelle. So don't just say prokaryotes don't have organelles. They do. It's just ribosomes aren't membrane-bound. Prokaryotes come in 3 different shapes: circles, rods, and spirals. Circles, rods, spirals. So if you've ever had like streptococcus, like strep throat, you were getting infected by a bacterial species that looks like circles. You can see here that here's a prokaryotic cell. This would be a coccus, of course, like a circle. Cytoplasm capsule, an advantage. Cell walls are important features. They help. Why are they important features? Here's what I would know. They help maintain shape, protection, of course. Hypotonic environment, which means water would want to enter, and so the cell would want to burst, but however, cell walls prevent that. Eukaryotic cell walls are cellulose or chitin. Bacterial cell walls are peptidoglycan. That is a common kind of question that you see, being able to contrast those two terms. The capsule is considered a virulence factor, which means it enhances the ability to cause disease. Virulence, a very virulent species would cause great disease. So the capsule protects cells from engulfment and actually helps to evade immune systems. It's an adaptation, an advantage. It increases fitness. The glycocalyx is a layer of polysaccharides. So this is a term that people, you don't really hear about this in 9th grade biology. So I would make a note card about this. This is a layer of polysaccharides on the outside of the cell wall. And it aids drying out, resisting the immune system. The capsule resists removal. And so this is an example of what the glycocalyx looks like right here. There's a lot of different factors on the outside that help a cell survive. Okay. Some bacteria, called cyanobacteria, actually have an internal membrane system. Cyanobacteria actually carry out photosynthesis. Some prokaryotes can move by flagella. That would be an advantage, right? Being able to avoid predators, find nutritious food sources, that would be an advantage. A flagella is a very complex system. It's actually a motor. We'd love to talk about irreducible complexity here and the argument for intelligent design, but you have to have these components for this thing to work correctly. And so it's just a really beautiful thing to see. I mean, engineered, right? Engineered. Flagella provide motility. Fimbriae are small bristle-like fibers that are used to attach. And then conjugation pili is used to pass DNA from one cell to another, which of course can lead to gene transfer. Cytoskeleton helps maintain shape like our skeleton does. You can answer these questions if you so desire. We kind of already talked about this one. Internal membranes. If you want to pause it right here and think like, what would be the advantage of having internal membranes inside of a cell? Pause. So the answer to that is, don't move that. The answer to that is surface area. You have more space to carry out reactions. And so the introduction to eukaryotic cells, again, we'll be practicing cells in class and stuff like that. But here's a cell. The cell has been taught as like a city, with a lot of different features of it. Like the Golgi is like the post office, and the nucleus is like City Hall, and the lysosome is like the recycling center, and all that's fair, and all that's true. But here's just, why are we learning this? This is the foundation of so many diseases. For example, Tay-Sachs disease. It's a lethal disease, completely lethal, 100% lethal, tragic disease. This young boy who didn't make it, he had Tay-Sachs disease, created in God's image, no longer suffering, believe that God does what is right, and with a child brings him into his heavenly kingdom. The Tay-Sachs is a disorder of the lysosomes based upon a hexa gene that doesn't make an enzyme correctly, which therefore builds up toxic chemicals in the brain. And so eukaryotes, again, there's lots of videos that you can see, crash course and all sorts of stuff like that. But we'll move on to endosymbiosis as we close out this video. You have to know, and we'll practice explaining it in class, endosymbiosis. And so endosymbiosis is how science, without a creator, explained how the mitochondria and chloroplast came to be. Okay? And then some people in the Christian faith would argue that, God would have allowed these materials to take place. I would ask your Bible teachers, and that I don't have time to get into that conversation right now. But nonetheless. All right, so here we go. Everything starts with a prokaryotic cell. All right, so the cell starts, creating membranes inside of it. Where did organelles and nucleus come from? So the nucleus kind of is gained by a lot of different, folding of the membrane. And then all of a sudden, this cell, this ancient cell, engulfs an aerobic bacterium. Okay? And it engulfs it first, because plants have mitochondria, right? And this aerobic bacterium is able to break down sugars in the presence of oxygen. This increases fitness of this cell. And therefore, instead of destroying it, the cell is like, well, wait, this is helping me. I have an advantage if I don't destroy it. And it's not thinking about this. It's just naturally, just kind of this cell survived because it had more energy. And so it led to greater success. In a similar way, a cell gains a photosynthetic bacterium. Okay, so engulfs it. And instead of, breaking it down, it has an advantage, and it's able to produce sugars. And so this is the ancestry of animal cells, and this is the ancestry of plant cells. And so here's just another kind of picture here. Nuclear envelope, nucleus forms, engulfs an aerobic heterotrophic prokaryote, which means using oxygen, breaking down sugars to generate ATP from that energy. And then that cell, in the plant lineage, engulfs a photosynthetic prokaryote and lets it start to survive and passes on. Again, we're talking billions of years here. Ancestral heterotrophic eukaryote. Heterotrophic means it has to eat something else different. Ancestral photosynthetic, which means it's making with light. And so the evidence for endosymbiosis, know this. This is a note card. Know this. Mad doctor. Membranes. Some organelles have double membranes. Okay? And, well, let's just go back here. Double membranes. Mitochondria and chloroplasts have double membranes. Antibiotics. They're susceptible to antibiotics, like other prokaryotes. Division. Mitochondria and chloroplasts reproduce, similar to prokaryotes. Mitochondria and chloroplasts have their own DNA. Mitochondria and chloroplasts also have their ribosomes, which are identical to prokaryotic ribosomes. Hey, that's good evidence, right? That's good evidence from this theory standpoint that, hey, did God allow this to happen this way? Or did he structure it this way? Good questions to think about, good questions to ask. Okay, and so eukaryotic cells are a lot more complex and specialized than prokaryotic cells. And they are compartmentalized. That's a big key word here, compartmentalization. They contain small structures called organelles. We talked about that. And a system that you need to know about is the endomembrane system. We'll work on this in class, but basically it starts with a nucleus, nuclear membrane, and then it goes to ER, and then the Golgi and vesicles. This allows organelles to communicate with each other and package and ship things to where they need to be, whether inside the cell or outside the cell. And then, of course, energy-related mitochondria and chloroplast. Mitochondria ATP powerhouse, chloroplast making food from sunlight. And here's a picture that you can kind of study, and here's a plant study you can kind of study. We'll practice in class. So I hope this was helpful. This is the first lecture. I've already done the organelle lecture, and we'll be practicing together questions in class. I'll be asking you questions, asking you to explain stuff maybe in teams. We'll try to do something different to learn a little differently while also trying to make up some time that we have missed, okay? So proud of y'all. So thankful for you. Hope you have a good rest of your day.